Good morning, students. Am I audible?
Morning, ma'am. Yes, ma'am, you're audible.
Okay, so I guess all the students know that instead of your IPR class, I'm taking class.
Yes, ma'am, we know.
Okay, fine, because I still see only 9 students in the meeting, 10 including me. So, and it's 10:19 already, right? Anyways, we'll start, right? So, we discussed fungi last time. The journal about the fungi, which I told you that you read about fungi journal yourself, and our main focus was on the type of the symptoms which are caused by fungi in the plant, right? So, can we just quickly revise about fungi, and then we'll start with today's topic. So, can you who will tell me what are the 3 basic types of the symptoms shown by fungi in the plants? 3 basic categories, right? फिर उन categories के अंदर further sub types है, but first 3 basic categories in which the symptoms are being categorized in plants which are caused by fungi. Hurry up, cool. Tell me, should I ask randomly or you're going to raise your hands? Yes, Ayushi. Devanshi can't speak today. Janvi, no. Namrata, नेहल, आप लोग पढ़ते नहीं हो बिल्कुल भी घर जाके, या क्लास में भी ध्यान से नहीं सुना है? हाँ, एटलीस्ट टेल मी समथिंग. Yes, Yukta, is your mic working now?
Hello.
Hello.
Yes, ma'am.
Yes. How do we categorize the symptoms which are caused by fungi?
The common symptoms that I know of are like we can observe the mosaic leaf patterns, the leaf spots,
Mm-hmm.
and we can also note like the stunting of plant,
Mm-hmm.
or
Stunting.
yellowing of leaves, or crinkled leaves.
Mm-hmm. Leaf cankers. Okay, fine. So you are telling about the symptoms which are caused. So nobody is going to tell me about the categories in which the symptoms are being categorized, right? Uh, okay, I'll just move ahead. So in my next class, if you're not able to answer what I'm asking, and first,
I know.
Namrata, okay, you know, then why didn't you raise your hand, Namrata?
Ma'am, I just joined. I just joined.
Okay, and you joined very late, at least 7 minutes late.
Yes.
That's not right. And next time in my class, I'll put that, slot also, that after 5 minutes, nobody will join without my permission. So make sure you all join on time. And it's already 10, not even 9 o'clock class that I expect you all to be, right, waking up and coming to the class directly. So please, huh? Okay, yes, Namrata, go for it.
You were asking about symptoms, ma'am.
I'm asking the categories basically. Which are right. When we studied the symptoms which are caused by fungi. So I told you that there are 3 major
Okay.
categories, right?
Yes, ma'am. Yes.
Yes.
Necrotic symptom.
Yes.
Then abnormal growth and development of plantation, and then the third one is the other symptoms.
Other symptoms. Okay, fine. Yes. So can anyone tell me about the necrotic symptoms? What are necrotic symptoms? What does necrotic means, and what kind of a symptoms come under this kind of a category? Yes, Nehal.
Ma'am, in necrotic symptom, basically the death of the tissue or the organs of the plant take place.
Yes, that's right. Okay. So can anyone tell me what is hypersensitive response or HR response? Hypersensitive. Yes,
Ma'am,
Shivani.
when when a response to a particular reaction or pathogen invasion gets more than enough, like
More than, yes. Yes, hypersensitive response is basically by the host in it is a kind of a we can say a defense mechanism in a host, that when some pathogen attack the host, they generate a hypersensitive response, and which leads to the dead tissues and death of those tissues where the pathogen has invaded, so that they can stop the spread of the pathogen to the healthy plant tissue or the other areas, right? So this is basically a defense mechanism which is also being shown by the plants in case of infection by certain plants pathogens, right? So we have a different kind of a leaf spots also, such as short holes. Then we studied about the blast, we studied cankers, we studied scab, we studied anthracnose, we studied blotch, we studied dieback, decline, damping off, basal stem rot, so many. And then second one was the abnormal, third one, abnormal growth and development, where we studied the galls, which are caused by certain kind of a bacteria or certain kind of a fungus. Galls are caused by the bacteria also. Then powdery scabs and the witch's broom, and the leaf curling symptoms, and other symptoms include the gumoses or vascular wilts and the wilts, right? And I told you last time the difference also, that what is the difference between the wilt which is caused by the pathogen and due to the overuse of a water or a shortage of a water. So can anyone tell me the difference, that what is the wilting in a plant which is being caused maybe overwatering, or due to the less water in the plant they become wilt, or wilts which are being caused by certain pathogens. So one basic difference was there, I told you last time. Do you remember it? No. So wilts which are caused by pathogens are irreversible. We cannot make them reversible. Like plant will not be okay, they'll be wilted and they'll die. But the wilts which are being caused by the maybe excess of a water or a less water can be reversible if a proper watering or after the situation becomes all right after some time. So this is the major difference that we studied, right? So today, again, in continuation, like we are talking about the plant pathogen in general and the symptoms. So we are going to discuss about the second pathogen, very important pathogen in the plant, which is bacteria. So lot of bacterial diseases cause lot of losses in the plant, and mostly the symptoms which are caused or shown by the bacteria, like cankers or galls, they are the similar kind which are being shown by the fungal diseases also, but still they are being categorized in a particular way, right? So if we talk about the bacteria, first we'll just go through the bacteria, that what is bacteria and its general characteristics, which you all know because you've studied micro 1 and micro 2 course. So I'm sure these things are not new to you, right? So we know then that bacteria are a single cellular microscopic organism, and they have been grouped into prokaryotes, which means absence of the nucleus, and they grow in numbers, not in size. So they make number of copies of themselves by dividing into half. There are three basic shapes of a bacteria: rod shaped, spherical, or a curved shaped spirali, or there are a lot of cocci, bacilli, and many other shapes which are there, in which they are commonly found. Then respirations can be anaerobic bacteria, which does not require oxygen for respiration. Then there are aerobic bacteria which require oxygen for respiration, right? Then how do we visualize these kind of a bacteria? So there's a gram staining, which is again on the name of the person who invented this training, that we stain the bacteria with a gram stain, and then they are being again categorized in a gram positive or a gram negative bacteria, right? So gram staining bacteria or gram staining is a method of differentiating bacterial species into two large groups, which are based on their chemical and physical property of their cell wall. So you know that what composition in the cell wall is differ, then they are being characterized as a gram positive or a gram negative. So gram positive bacteria, those bacteria when they are stained in a gram stain result in a purple color, and gram negative bacteria, those bacteria when they are stained in a gram stain results in a pink color. They are called gram negative bacteria. So bacteria can move by using cilia and flagella. They basically move by using these, And nutrition, if we talk about, there are like autotrophic bacteria or a heterotrophic or symbiotic or a parasitic bacteria. So autotrophic, as the name, these are able to synthesize their own food, and heterotrophic, they are unable to synthesize their own food. They depend on the organic material such as saprophytic bacteria. These feed on the dead and decaying. Then there are certain symbiotic bacteria also. That means they gain a mutual benefit from other organisms. Nitrogen fixing bacteria or rhizobium come under this group. Then parasitic bacteria. Now these are the bacteria which when present in a plants, animals and humans, they feed on the host cell. They cause diseases or a harm to the host cell. So most bacteria, they fall under this category which cause diseases. So these are the again different nutritional type, chemical, carbon sources, or what kind of a different chemoautotroph, chemoheterotroph, photoheterotroph, right? So just go through this. Then cell wall, we know that cell wall is made up of a glycoproteinurine, and what is the main function? They provide support and mechanical strength and rigidity to the cell, and it also protects cell from bursting in a hypotonic medium. Then a common bacterial cell wall material is peptidoglycan, which is made from polysaccharide chain cross-linked by peptides containing D amino acids. And their cell walls are different from the cell wall of a fungi, which are made up of a cellulose or a chitin. So they differ, right? And again, cell wall is essential to the survival of many bacteria. And again, the antibiotic penicillin is able to kill bacteria by inhibiting a step in the synthesis of peptidoglycan. So that is how the antibiotics, they also act here. Then again, they are broadly speaking two different types of cell wall in bacteria, like thick one in a gram positive and thinner one in a gram negative. So this is the basis for a gram stain also. Then okay, so I'll just go through this. Gram positive bacteria has a thick cell wall with a many layer of peptidoglycan, and gram negative bacteria, they have a thin cell wall with a few layers of a peptidoglycan, which is surrounded by a second lipid membrane containing lipopolysaccharides and a lipoprotein. Then they have a plasma membrane also, so it is known as a cytoplasmic membrane or a cell membrane. Again, it is composed of a phospholipids, proteins, and carbohydrates. It helps in a transportation of substances and also removal of waste from the body. Again, it helps in providing a mechanical barrier to the cell, right? It acts as a semi-permeable membrane, which allows the movement of a selected material to inside and outside of the cell. So that is why it is known as a semi-permeable membrane. Cytoplasm helps in the cellular growth, metabolism, and replication. It is again storehouse of different chemicals and components which are used or which are important to sustain the life of a bacterium. Ribosomes, they have a ribosomes. We all know this. These are the place where the protein is being synthesized. Then plasmids, again, they are small circular circle of a DNA. And bacterial cells, they have many plasmids. They are used to exchange DNA between the bacterial cell. Flagella is for a movement. This is a rigid rotating tail. It helps the cell to move in a clockwise and clockwise forward, and also helps the cells to spin. So this is helped in the locomotion of a bacteria. Villi are again short protein appendages. These are smaller than flagella, and it fixes bacteria to the surfaces. It helps in a reproduction during the conjugation. Then again, capsule is a slime layer which covers the outside of the cell wall, and it is composed of a polysaccharide. So it is used to stick cells together and work as a food reserve. It protects the cell from drying and from other chemicals, right? Reproduction is by cell division, binary fission, and sometimes they also reproduce by budding, right? Endospores, again, these are the common structures which are formed. So these are dormant, tough, and non-reproductive structures which are produced by certain bacteria from Firmicute phylum, and it is a dormant form to which bacterium can reduce itself. And endosperm formation is usually during the unfavorable conditions when there are less nutrients, right? So in those conditions, this endosperm formation takes place, And during endosperm formation, bacteria, it divide within its cell wall one side, then engulfs the other side. So endospore enable bacteria to lie dormant for extended periods, for even centuries. So whenever they'll get a favorable conditions or a favorable environment, they can again multiply. So example of plant pathogenic bacteria are Erwinia, Mylovora, Xanthomonas campestris, Pseudomonas syringes, Xanthomonas oryzae, or Agrobacterium rhizogens. So these are just few examples which cause diseases in plants. There are a number of bacteria which cause a lot of diseases in the plants. So when we talk about the symptoms, because symptoms are important in plants, because plants cannot speak for themselves. They cannot tell anyone that we are not well, so come and give us some medicine. This is not the case, because when as a human or we become sick, so we can tell that okay, this is not fine, so we should go and see a doctor. So this is not the case of the plant. So definitely we have to see that, right? We have to see what are the changes in the plants. So that can be seen or understood by the first thing, symptoms, right? Visual symptoms on the plants which can be caused due to any kind of a pathogen, right? So symptoms of bacterial infection, in again, they are much like symptoms in a fungal diseases, as I told you in the beginning. Like um, cankers are caused by the fungal pathogens also, they are caused by bacterial pathogens also. Scabs are caused by fungal pathogens also, they are caused by bacterial pathogens also. Wilts are caused by fungal pathogens also, and there are bacterial wilts also. Galls are caused by fungal pathogens also, and galls are caused by bacterial pathogens also. So there are a number of symptoms which are similar, such as leaf spots, blights, wilts, scabs, cankers, soft rots of a root, storage organs and fruits, and overgrowth. So these are the typical similar, usually similar symptoms, right? So bacterial spots, when we talk about, again, they appear on the leaves or could be on the fruits also, and could be on the stems also. So if spots appear and advance rapidly, the disease is considered a blight, right? So that means if spots, they increase rapidly, number of spots, they increase rapidly within a period of a time, so that disease can be considered as a blight. And in my last lecture, I told you that blight means it gives a burned appearance to the leaf, right? So number of leaf spots on the leaves, they combine to form kind of a elongated spots which give a burned up, as if it gives us the impression that leaf is burned, right? So if spots on leaves of a dicot plant often have a rotten or a fishy odor, and are water soaked, and are initially confined between the leaf veins, and sometimes they'll appear angular in shape. Then in some cases, bacterial ooze. So this also I told you that bacterial ooze is a sign of a bacterial disease. So if we just cut open, or we just cut the twig of any plant which we feel is infected by certain bacteria, it will emerge ooze like substance, will be a white color, or some that kind of a substance can be seen. So that is called as a bacterial ooze. Again, this is diagnostic for bacterial infections. Sometime chlorotic halo will surround the bacterial lesion of an infected leaves, and spots may coalesce, causing large area of necrotic tissue. So they may overlap, they may combine different spots, they may combine and form a large areas of a necrotic tissue. Again, bacterial spots will appear as a streaks or a stripes on the monocot plants. Streaks in a monocot plant के leaves कैसे होते हैं? They are thin and they are long, right? Monocot plants में, dicot में leaves are broad, right? So monocot plant में जब spots appear होंगे, they'll appear as a streaks, right? Or a stripes in a longer form, elongated forms. So almost all bacterial leaf spots and blights are caused by genera Pseudomonas and Xanthomonas. So they cause the bacterial leaf spot diseases. Fungal leaf spots हमने पढ़ा था Alternaria, Septoria, Phylosticta. So these called fungal leaf spots. Bacterial leaf spots are caused by Pseudomonas and Xanthomonas. These are Pseudomonas leaf spots being shown on certain leaf. Then cankers. I told you the cankers, they were a bacterial cankers also, and the cankers are caused by, especially the Pseudomonas and Xanthomonas, they cause canker disease of a stone fruits, right? And pome fruit trees and canker disease of a citrus respectively. Canker symptoms again can be seen on a They form a corky growth on the plant leaves, stems, twigs, and branches, and even on the fruits. The most important symptom of bacterial canker disease in stone fruit and a pome fruit is development of cankers and gum exudation. That means the gum, it comes out of a tree due to the canker. When we talk about a stone fruit and a pome fruit, so in a pome fruit, there are trees such as apple, right? Apple and pear, these all can come under pome fruits. And when we talk about stone fruits, there are fruits such as plum, pear, peach, cherry. All these are stone fruits because इनका जो एंडोकार्प होता है वो स्टोनी होता है. अंदर आप खा के देखते हो तो बड़ा स्टोनी एंडोकार्प होता है. So that is why they are called as a stone fruits, right? So cankers can be slightly sunken. संकन का मतलब होता है डीप, समवेयर अंदर. हम उसको देखेंगे स्टेम पे तो डीप हमें नजर पे आएगा. इट विल बी सीन इन डीप समवेयर इनसाइड द स्टेम. Dark brown or much longer than a broad. The cortical tissue of canker can be orange brown or to dark brown in a color. Gum is produced in most cankers and some branches and twigs. Cankers do not produce gum. They may have a sour odor and be soft, sunken, and moist. Cankers that girdle trunks and branches can result in a leaf stress and eventual dieback of the portion of the tree distal to the canker. So this is the outcome of the canker which are caused by the bacterial pathogens. Again, the citrus canker on a leaf is seen like that. On a fruit, you can see it is seen on like this. These are the initial and these are the later symptoms, can be seen from light orange color to the dark color in the later stages. Then bacterial galls. So bacterial galls can be produced by genus Acrobacterium and Arthrobacter, Pseudomonas, Rhizobacter, and Rhodococcus species of bacteria. Agrobacterium tumefaciens, Agrobacterium rubi, and Agrobacterium vitis alone are responsible for galls in around 390 plant genera. Agrobacterium tumefaciens cause a lot of galls in the plants. Galls of these genera are crown gall, crown knot, root knot, and root galls. These are known as by different names, right? So species of these bacteria are thought to be present in the most agricultural soil, and a wound in the host is required. So bacterial pathogens cannot get entry to the plant unless there is some kind of a wound, right? So that wound is required to gain entry into the host tissue. And gall tissue is composed of disorganized and proliferating cells that are multiplying or dividing, that multiply in intercellular spaces in the vicinity of the wound. In the presence of the pathogen, rapid and continuous cell division of the plant tissue take place. Gall damage can be benign to deadly, and crown gall first appears as a small whitish soft round overgrowths on the plant crown or at the main root. But then again, the color of the galls, which are caused by Agrobacterium tumefaciens, can be orange brown, and it then with a period of time it becomes large, it enlarges, and surface can become convoluted and dark brown in color. So this is most often found in the commercial nurseries where plants are being grown for selling purposes. So this is again a crown gall. Here you can see the crown gall. Here also some berry, you can see the crown gall. So you see the stem. This is so thin, but due to the formation of a gall, such a big extra growth can be seen here, right? Then bacterial vascular base, right? So we know the vascular tissues, we all know in the plant xylem and phloem, right? Vascular tissues. So vascular beds caused by bacteria, they affect herbaceous plants such as vegetables, field crops, ornamentals, and some tropical plants also. So the pathogen, they enters, they multiply and moves through the xylem vessels. So basically vascular wilts when we talk about, so pathogen is somewhere affecting the vascular tissues, xylem vessels, right? Vessels of the host plant, and interferes with the translocation of nutrients and water by producing gum. So due to which the wilting take place. The pathogen will often again destroy the part of cell walls of the xylem vessels, which will again result in a pocket of bacteria, gums, and a cellular debris in that particular area. And the symptoms of bacterial wilt disease includes wilting and death of the above ground part of the plant. Wilting hota hai drooping of the plant, right? Sometimes in the field we see the some plants are just literally dying and lying somewhere near the ground surface. Whether sometimes whether whereas the all other plants are very healthy, so that can be a symptom of a wilt. And when wilt is caused due to the water stress or something, so that means that kind of a symptom should be seen in all the plants in that field. If it is being caused by certain bacteria and fungus, that means only few plants can be seen in that condition, right? So that's how we can differentiate here also. And in some cases, bacterial ooze seeps out through stomata or cracks onto the surface of infected leaves. And usually this ooze does not occur until the infected plant tissue is dead. So only after the tissue is dead, then we can see this ooze. So this is again the bacterial wilt. This toward this side, this seems to be a tomato bacterial wilt, and this is again some plant wilt which is caused by the bacteria. Then we have a bacterial soft rots also, right? We last time we discussed the soft rots caused by the fungus, and here there are bacterial soft rots. So soft rots, that means the tissue becomes soft due to the rotting which is being caused by the bacteria, right? So primarily the bacteria that cause soft rots in a living plant tissue include Erwinia species, Pseudomonas, Bacillus, and Clostridium species. And sometimes these are caused by non-pathogenic phytopathogenic bacteria also, which are saprophytes that grow in tissue that has been killed by pathogenic or environmental causes. So it could be due to the saprophytes also. Soft rots attack a number of hosts and are best known for causing disease in a fleshy part structures both above and below ground. So fruits में ज़्यादा इस तरह के rots नजर आएंगे हमें, right? So these bacteria are almost always present where susceptible plant under stress are in the field or in storage. Soft rot pathogens, they enter the host through the wounds also. And yes, so after entering the host tissue, these bacteria, they produce enzymes so that break down the middle lamella, which cause separation of the cells at the site of the infection. So when these cells die and they disintegrate, the rotting tissue, they becomes watery and soft, and bacteria will form a slimy, foul-smelling ooze that will ooze out or come out of the infected tissue. So bacterial ooze is again a diagnostic for a soft rot diseases. So this kind of ooze or a smelly ooze can be seen in the plants or plant parts. So this is again different soft rots. We can see first one is a potato, and then again this is one is a tomato. This one seems to be a capsicum, right? And this is the apple somewhere. So these are the different soft rots which are being caused by the bacteria. Then we have a bacterial scabs also. So the scabs basically infect underground part of the or below ground part of the plant, such as potatoes. And common scab of potato is caused by Streptomyces scabies, which causes localized scabby lesions on the outer surface of the tuber. And corky tissue will form below and around the lesion which is being formed. So rot pathogen can gain entrance into the host tissue through these lesions and then either further disintegrate or degrade the host further. This is again example of a potato scab which is caused by Streptomyces scabi. So these kind of symptoms on the skin of the potato we can see due to the scab which is being caused by the bacterial pathogens, right? So these are the common bacterial symptoms which are being shown. See, in the beginning, I told you that the symptoms are usually similar, caused by the fungi and bacteria. So how do you remember this? How do you differentiate and remember this, right? So the easiest way is just you form it in a tabular form, right? In one side, you write a symptom which is caused by fungus, in another side, bacteria, and in numbering side, you write down the type of the symptom. तो इसमें क्या होगा कि you'll be able to see the difference and similarity and you'll be able to remember easily. So this is the best way to remember the symptoms which are caused by bacteria and bacterial and fungal pathogens, right? So this is all from my side. If there's any question from you, please, this is open for the questions. Yes, the hand is raised. Manaswani. Yes, Manaswani.
Ma'am, if the symptoms between, for bacterial and fungal infections are so similar, how would we like identify if it's a bacterial or a fungal infection?
Yeah, I'll tell you. The Manaswani symptoms. This is good question, right? Definitely that we should know that. So Manaswani, the first step in a fungal pathogen, what we do is usually we take a sample. We take the sample. We get it into the lab. We just cut the sections of those infected parts and we see it under the microscope. So as I told you, in fungal pathogens we are definitely going to see some kind of a spores or some kind of a fruiting body or some kind of a structure. So that will tell us that this is a fungal pathogen. Similarly, in case of a bacterial pathogen also, we can in the beginning definitely will not know, right? We need to do certain diagnosis or a preliminary diagnosis which will delineate the causes. And second one thing, as I told you, the bacterial ooze is, or bacterial streaming, it is called as a streaming also. So this is the best diagnostic symptom which can differentiate between the bacterial pathogen and a fungal pathogen. जब fungal causes होगा तो bacterial ooze हमें नजर नहीं आए. In the practical test I'm going to demonstrate you this, that bacterial ooze कैसे नजर आता है. So when the fungal pathogen is there, we are not going to see that bacterial ooze, whereas in case of a bacterial pathogen, this bacterial ooze will be the common symptom that we are going to see. So this is the first difference, but still in the lab for fungal, initially we'll see it under the microscope and we'll try to see the fruiting bodies, and for bacterial pathogens we'll try to isolate it by different methods, and after then certain chemical testing is being performed to identify the bacterial pathogen. So this is how it differentiates. Like in the beginning we cannot say anything, but just on the basis of the science we can say that yes, this is a bacteria and this is a fungal. But again, then diagnosis is to be required to be done in the lab, and before those diagnoses, nothing can be confirmed. Okay.
Okay, ma'am. Thank you.
Okay, Manaswani. So others, please just try to see, be attentive in the class. It is not at all difficult. It is something new that you're going to read, and it is interesting, right? How pathogen causes infection. So and after the class, when you go home, or after the class, when you sit, just try to go through the things and just try to read little extra about. In the internet you'll find 100,000 of things. Just try to read little extra about what I have already taught you. So that will be all for your this particular course, right? I can say that, right? So okay, so since I'll paste one assignment question as usual, and then we can discuss that question also. Okay, I've assigned just one in Microsoft from two small questions. So just quickly do it and back. Is it visible or not?
No, ma'am.
Just a moment, it should be visible. It should be visible. Is it? Not yet.
No, ma'am, not yet.
Just a moment, just a moment. Is it visible? Oh shit, I did it in a wrong. Just a moment, I did it in a wrong section, I guess. Just a moment. Just a moment, I pasted in a wrong group actually. Examination help desk open था मैंने उसमें डाल दिया. Just a moment. Okay, this is our pathology. It should be visible now, I guess.
Yes, ma'am, it is.
Okay, you guys can submit this quiz till 11:30, I guess. So in the meanwhile, we can take the attendance also. Please switch on your cameras for attendance. Adhish Mishra,
Present, ma'am.
Ayushi Jain,
Present, ma'am.
Sam,
Present, ma'am.
Ivanshi, Ishita,
Present, ma'am.
okay, Ishita, Janvi Singh, Manaswani Sridhar,
Present, ma'am.
नंबर मानसवनी आपको देख के इतना रिलैक्स फील होता है यू आर इन द अदर साइड ऑफ इंडिया एंड वेयर देयर इज नो कोल्ड एंड you see the children here all must be in lot of clothes right so much cold in North India right now.
Yes, ma'am. It suddenly got like pretty hot here, so
Oh.
like I'm very comfortable. I don't
yeah
like the
yeah
cold.
yeah that's why when I see you and when I see other so it is a lot of difference right.
Yeah.
yeah okay Namrata,
Present, ma'am.
Nandini Raj,
Present, ma'am.
Nehal Joshi,
Present, ma'am.
Rupal Joshi,
Present, ma'am.
Sahiba,
Present, ma'am.
Sanjana,
Present, ma'am.
Shakti,
Present, ma'am.
Shivani, Tanvi,
Present, ma'am.
Ridev,
Present, ma'am.
and then we have Vivek, Yukta, Yuvraj.
Present, ma'am.
Okay. Okay. Vivek, I don't hear you. So I really hope आप लोग अपने घर वापस चले गए हो कि अभी भी पीजी में हैं कुछ स्टूडेंट. Are you all
Yes,
gone
ma'am.
back to your gone home? Okay, fine.
No, ma'am.
Okay, great. So Athoiba is still not there. So Adhish, can you please contact Athoiba and see whether he's still in PG or he's gone back? I don't know.
Okay, ma'am. Most probably he has went back.
He's went back. Okay, but he's not attending classes now. So I was little worried.
Yes,
Anyways,
ma'am.
please just
I'll talk with him.
yeah, please just try to inform him that don't miss the classes. There are some extra classes are also being published since this is a fast track batch. So he'll lose lot of attendance, right? Okay,
Yes,
okay,
ma'am.
students. So I'll see you tomorrow in the class. Thank you so much.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.